قريبه قوي، كانت نزلت في 2012 وكانت هاتت ان وقف عند البريماتيك ديفيجن، ما حصلش ديفيجن في السبيرم بس كانت جايه في المرض. لكن دي ترى ممكن ده يبقى هو جامد قوي الناس عندهم.
لا الحاجه دي في المحاضره بتاعتي في كلام لو تحب ان انا نكمل او انا وراها او اي
حاجه. يا ريت، انا شايف ان احنا ايه نخلي مصطفى يتكلم عن
الدسكشن في الاخر
بحيث ان سخنه على طول.
ماشي اتفضل.
دكتور مصطفى سعيد ستيم سيل الميل ريبرودكشن عشان نكمل التوك بتاعت محمد جلال. هو كان المفروض ان ايمن مسام مصطفى ده
انا عشان اخش في سكه ثانيه.
مصطفى استاذ مساعد قسم الامراض الجلديه. تباركوا له بقى، اترقى الاسبوع اللي فات.
بسم الله الرحمن الرحيم. My talk is stem cells on the way to acute shutdown, or stem cell and non-obstructive azospermia. We'll start some clinical overview, then we'll go a little bit in basics, then go again to a possible clinical. So we'll say that male factor is responsible for 50% of infertile couple, and the azospermia represent from 1 to 10% of infertile men in the general population, and up to 15 among infertile men. Non-obstructive azospermia is defined as characterized by small volume testes and elevated follicular stimulating hormone with severely deficient spermatogenesis. The etiology is multifactorial, ranging from idiopathic, like sertoli cell-only syndrome, genetics like Klinefelter, hormonal hypogonadotrophic hypogonadism, undescended testes, hyperhyalinization, chemotherapy, infection, injuries, varicocele, tumor, and mixed pathology. The story of spermatogonial stem cell start from a little bit. We'll go back to Dr. Muhammad talk. The germ cells are derived from a population of primordial germ cells. From primordial germ cells that rise from the proximal epiblast of the embryo, then migrate to the primitive gonads, where it differentiates to either sperm or oocyte. Here we speak about sperm. Once primordial germ cells arrive the genital reach, they are enclosed by somatic stem cells and become gonocytes. These cells proliferate for a few days and then arrest in the G0, G1 phase of the cell cycle during fetal development. After birth, primordial germ cells migrate to the basement membrane of the seminiferous tubules and become spermatogonial stem cells. The spermatogonial stem cells are fundamental for the spermatogenesis process and have the ability to self-renewal to generate differentiated germ cells. Only 1% of the cells in the seminiferous tubules represent the stem cell, spermatogonial stem cells. Here is showing that this is the primordial germ cells. It's migrates to 6 differentiated. This is the basal lamina, basement membrane where they lie. This is the spermatogonial stem cells, and this is the process: first meiosis, second meiosis. The spermatogonial stem cell divide asymmetrically to give rise to one stem cell and to other cell, which will complete the differentiation up to sperm. This is the picture of the testis with the seminiferous tubules. This is the basement membrane. This is spermatogonial, the primary spermatocytes, secondary spermatocytes, early spermatid, and the sperm. This is the photograph which showing the detailed process of spermatogenesis, in which spermatogonial stem cell give rise to type A and type B. Type A stay as reserve cell to form further renewal, and type B completes the process where Dr. Muhammad said through first meiosis. The primary spermatocyte, before entering the first meiosis, they duplicate the DNA, so it's called 4N or tetraploid cell. First meiosis become 2N. Secondary spermatocyte, in second meiosis, giving secondary spermatocyte, and finally spermiation with morphogenesis of the sperm, giving rise to the highly specialized, complicated living organ, which is the sperm. I thought it is like a living organ. Okay. So we have also what's called spermatogonial stem cell niche. Niche means the microenvironment in which the cells is lying. It's composed of cells, growth factors, and other factors. In here in the testis, we have mainly the seminiferous, mainly the Sertoli cell. We have also the glial cell line derived neurotrophic factor, which is essential for the self-renewal and survival of spermatogonial stem cell. This is a picture showing the cell niche of spermatogonial, in which there is myoid cell in the basement membrane, also choline-stimulating factor 1, also glial-derived neurotrophic factor. The Sertoli cell have a big role, Leydig cells with its secretion of testosterone and also other unidentified factors. This photograph showing how complicated the process of signaling to the cell to either go through becoming a stem cell to have the ability further for self-renewal, or either to differentiation. We have many signaling pathways, including bone morphogenetic protein 4, glial-derived neurotrophic factor, stem cell factor, and other factors. This table showing the role of different factors which play a role in differentiation and proliferation of the stem cell. Every cell has a marker. The marker for stem cell to be identified, we have many markers, especially this marker, which is alpha-6 integrin. Previously it was named, now it's called CD49F, beta-1 integrin, C1, CD9, and A-cadherin, CD24. This photograph showing the different markers of different cells. Stem cell markers, OCT4 and other many, progenitor and differentiated cell marker. What now we'll go to what breakthroughs on the way of spermatogonial stem cells, the major events which lead us to the discovery and the process of trying to develop a human sperm from these cells. In 1994, Prelister and Zimmermann, German, demonstrated the presence of a stem cell population responsible for continuous sperm production in mice. What he did, he take a biopsy from a fertile mouse, make dispersion of the cell, and transfer the stem cells to the bisulfan infertile induced mouse. And after 3 months, they demonstrated the presence of a sperm from that mouse to which we transfer the stem cell. In 2003, Toyoka et al. Showed that embryonic stem cells could form male germ cells when transplanted into reconstituted testicular tubules in vitro. Also, this embryonic stem cell derived embryoid bodies, which is a composite cells, a group of cells formed during the culture of embryonic stem cell, supported maturation of primordial germ cells into haploid male gametes, which when injected into oocytes restored the somatic diploid chromosome complement and developed into blastocyst. This also in animal. Also in 2004, in vitro generation of a germ cell line. This is Nairenia. In 2004, they generate a stem germ stem cell from a pluripotent teratocarcinoma cells, and formation of mature seminiferous tubule structures, and support of spermatogenesis after transplantation into recipient testes. In 2006, it was a big throw in which in vitro differentiated embryonic stem cells give rise to male gametes that can generate offspring in mice. This is also Nairenia et al. In 2006. What they did, they make a detailed protocol for the in vitro generation of germ cells from urine embryonic stem cells. And they were able to generate offspring mice. They developed the embryonic stem cell into fully matured sperm. Not only this, they make ICSI of that sperm cells and the oocyte of the mouse and generate offspring in the mice with blastocyst formation in 20% of these mice. In 2007, this was a putative human male germ cells from bone marrow stem cell. Here, they use also in animals, they use the bone marrow stem cell transplanted into the testis of a busulfan-treated infertility mouse model, appear to differentiate into germ cell, Sertoli cell, and the Leydig cells. So they form a composite seminiferous tubules from embryonic bone marrow stem cell. Conrad et al. In 2008 reported the isolation of spermatogonium stem cell from adult testicular tissue after generating human adult germ-line stem cell. This spermatogonium stem cell showed similar cellular and molecular characteristics to embryonic stem cell and germinal. They are pluripotent, however, they form teratoma when injected into immunosuppressed mice.
عايز ميه.
شكرا. However, some found that this human adult germ-line stem cell and fibroblast have a similar gene expression profile. Therefore, still this pluripotency of these cells is currently under debate. In 2009, there was a big throw which Narinia et al. Claimed that in vitro derivation of human sperm from embryonic stem cells. They are in Newcastle in UK. This paper was online a hit, but when I go through, they claim that the results clearly demonstrated that germ cells derived from XY human, it was the first time to say that human embryonic stem cell undergo meiotic division and produce haploid sperm-like cells. Unfortunately, this article was withdrawn from stem cell and the development. They said that there's some unscientific part in the process because there was no reproducibility of this and many defy against them. Now we go to some application of what we want to do. So preservation of fertility in prepubertal boys treated for cancer. From this background, we can say that it will be possible to freeze the spermatogonium stem cell collected from the testis by means of a testicular biopsy before they receive cancer treatment to restore fertility during their adult life after autologous transplantation. This is the photograph. The story starts, it's small child, prepubertal, will take a biopsy from, it's already immature, and it's known that the immature testes have a percent, we said that the spermatogonium stem cell represent 1% of the seminiferous tubule cells. However, in prepubertal testes, this percentage is more than adult testes. So we'll keep the testicular biopsy in liquid nitrogen cylinder, maybe up to 25 years, as Dr. said yesterday, and then used after receiving the treatment later in their life. Sadri, Arikan Aldikanai, this is Turkish, and colleagues reported in this case, the problem is that the yield of the spermatogonium stem cell is few. However, its percentage is few. So some claim that they can enhance the number of spermatogonium cells up to 18,000-fold increase over 64 days in their post-pubertal human germ-line stem cell, and this was in Journal of American Medicine in 2009. However, there is limitation. It remains unknown if prepubertal human spermatogonium stem cell will react in a similar fashion to the post-pubertal, and also if clonally expanded cells will repopulate the testis in the same fashion as de novo cells directly from the testis. Autologous spermatogonium stem cell transplantation, so now we have no rejection because we take the same patient spermatogonium stem cell. To a treated cancer patient harbors the theoretical risk of also reintroducing malignant cells, and this happened in the rat model in leukemia because this cell transferred to the mice of leukemia model, it make leukemia in the recipient mice. Now we come to another era of stem cells, which induce the pluripotent stem cells and non-obstructive azospermia. In a simple idea is that they introduce a set of transcription factors related to pluripotency can directly reprogram human somatic cells to produce induced pluripotent cells. These cells derived from testicular can be derived from testicular tissue and might be a potential source of cell-based therapies to treat some cases of male infertility. And Kobayashi from Japan successfully obtained induced pluripotent stem cells derived from the testicular tissue of men with Klinefelter syndrome. This is a very big, all of us know as andrologist or non-andrologist, Klinefelter syndrome is it is not so rare in the population. And the testis is severely dangerous spermatogenesis. He make an induced pluripotent cell from their testicular tissue by introducing 4 transcription factors, which OCT4 or octamer4, SOX2, KLF, and CMI using the lentiviral vector system. This is a big throw really, and they show that the marking of different transcription factors in the cells of induced pluripotent from Klinefelter syndrome. The morphology, proliferation, surface markers, and gene expression of these cells were similar to those of human embryonic stem cell. However, there is limitation also. The limitation is that in induced pluripotent stem cells, the chromosomal abnormalities seems to appear earlier in their culture. High frequency of mutations and a greater number of novel copy number variants are also present in induced pluripotent stem cells. Also, apparent DNA methylation and the retention of epigenetic markers from the cell of origin. And lastly, the lack of genetic stability in induced pluripotent stem cells precludes their use in humans. We come also to another story, where all of us know that genetic factors have a big role in male infertility. And the deletion of azospermia factor gene is one of the most common causes of genetic infertility in the male. They make a model for rat for that is deleted, and they make transfer of embryonic stem cell to that rat to enhance its fertility. As we know, as I told, that DAS-like is a master gene controlling germ cell differentiation. Ectopic expression of DAS-like gene promotes the dynamic differentiation of mouse embryonic stem cells into gametes in vitro. And they report that as few as 50,000 donor spermatogonia transplanted into only a single testis recipient effectively restored fecundity to detect deleted in azospermia-like deficient rats, yielding 100% germline transmission to progeny by natural mating. Here there is merge between using embryonic stem cell or spermatogonial stem cell and gene therapy. Newly, in 2011, in the Stem Cell Studies Journal, Abdelaziz Itan, they are the group from Cairo University. One of them is Dr. Taimur Mostafa. In 2011, they have provided evidence that in absence of any spermatogonial stem cell in the testis of the patient, this was in mice, they use the bone marrow-derived mesenchymal stem cell, and they make it to have the potential for in vitro transdifferentiation into germ cells and in vivo transdifferentiation into spermatids. This is a big breakthrough. Differentiated into spermatids and spermatocytes in the seminiferous tubules of azospermic rats. This was in 2011. Sertoli cell syndrome, as we know, only the Sertoli cell are there in the seminiferous tubules and no germ cells. What they do, in a special culture medium, germline stem cells, like cells, was found in 30% of the non-obstructive patient diagnosed with Sertoli cell syndrome. Myself, my MD thesis was using DNA flow cytometry in infertile azospermia testicular biopsy. Really, this was the percentage. In 25% of Sertoli cell syndrome, we found a peak. Flow cytometry gives peaks of DNA, 1N, 2N, and 4N. In my thesis, we found that 25% of the Sertoli cell only diagnosed patient have haploid cells. And this may have some reflection on this. The identification of such cases should prove more information for the application in assisted reproductive techniques. And in September 2012, there is a paper in the Cell Reports, direct differentiation of human pluripotent stem cells into haploid spermatogenic cells. And this is still there, this is not withdrawn. Male male pluripotent stem cells have the ability to differentiate directly into advanced germ cells and may represent a novel strategy for studying spermatogenesis in vivo. What they do, the human embryonic stem cell and the human-induced pluripotent stem cells from patients with Sertoli syndrome differentiate directly into advanced male germ cells, including post-meiotic spermatid-like cells in vitro without genetic manipulation. These cells was evidenced that they are haploid by expression of acrosine, transition protein 1, and protamine 1, which are uniquely found in spermatids and or sperm, as Dr. Gaballa said. Again, in September 2012, Repoledi in Fertility Sterility Journal, they published a paper saying in vitro production of haploid cells after co-culture of CD49F, this was before alpha integrin 4, with Sertoli cells from testicular sperm extraction in non-obstructive azospermic patient. This is also a big throw after the claim of Nairenia attack. They make marking of Sertoli cells with positive stem cell factor, recombinant GDNF, William tumor gene, GATA, and vimentin, with the presence of tight junction and the lipid droplets shown by orate staining, and the presence of haploid cells on day 5 of the co-culture was confirmed by fluorescent in situ hybridization analysis of chromosome 13, 18, 21, X, and Y. And this is really a big breakthrough in which they develop evidenced human haploid spermatid or sperm in vitro. So in vitro co-culture of spermatogonial stem cell from TZ sample, TZ means testicular sperm extraction of non-obstructive azospermic patient, along with Sertoli cells promoted meiosis induction and resulted in haploid cell generation. These results improve the existing protocols to generate spermatogenesis in vitro and open new ways for clinical translation in azospermic patients. And the hope that one day we will be able to grow spermatogonial stem cells to a human sperm on the way to a future death. So more basic research, transitional and clinical research is required before in vivo or in vitro maturation of germ cells can be applied for human clinical use. And will not say that it is nothing, that stem cell and 50 years of research will fix the problem. Inshallah, we hope so. Thank you very much.
شكرا دكتور مصطفى. تحب تخلي الدسكشن في الاخر ولا خليها في الاخر؟